Jai Hind everyone, I am Pankaj Kumar and today's class is about the calculations which we are doing in your class during your experiment in biological labs or in the chemistry labs. So it's a very basics and most fundamental things to be learned just before the going into the lab and experimenting anything. So here we go. The topic, as I mentioned here, biology labs solution makings. So it's very important. Let's see. What is mole? If you know, that's good. If you don't know, just remember it. It's a number. So that number is called is as a Avogadro number. What it is called? It's called as a Avogadro number, and its value is around 6.022 into 10 power 23 is the value of a mole. So if I'm saying a mole means one mole can be anything which will be equal to this much of number. If I'm saying a 1 mole of human being, so it may be this much of humans being will be there. If I am saying a mole of galaxies, so there will be so many things are there. This number of galaxies can be there. If I am saying a mole of rice, so if I am saying a mole of rice means there will be 6.022 into 10 power 23 rice seeds. Okay, so it's about the mole only. It's about numbers. So it is similar to the pair or the dozen, as we used to use this term. So pair is what? It is only a two shoes. If I will say is dozen, so what does it mean? Dozen means something which will be 12 in number. If I am saying 3 dozens, what does it mean? The 12 into 3, it means 36 dozens are there. Suppose if I am saying 3 pairs, what you will do? If I am saying 3 pairs means what you will do? 3 pair means 2 into 3 you will do. It means total 6 shoes will be there. So it's about what? It's just a number. So if just before going to the further topics, we have to understand how we can calculate the molecular mass and what does it mean molecular mass. So molecular mass is the weight of a molecule. It's a not exactly weight. Weight is not a proper term. Molecular mass is a mass of a molecule. So H2O. So how many hydrogen atoms are there? So there are two hydrogen atoms. So you just multiply with one. So mass of hydrogen is 1 into 2. I took 2 because there are two hydrogen. And the mass of the oxygen is how much? 16. So 1 into 2 plus 16 will be 18. So at 18 is the molecular mass of the H2O. Now see how we can calculate the mole of 25 gram of the water. So how you can calculate the mole of 25 gram of water? So what you have to do, you have to just divide the grams given mass by the molecular mass. So 1 mole of water contain of 18 gram of molecules. Okay, 18 gram of molecule. 1 mole of water contain 18 gram of molecules. So 25 by 18 means 1.39 is the molecular mass of 24 is the mole of 25 gram of water. So like same, you have to tell me the mole of NOH, KOH, CaCO3 and NS3. You just have to pause your button and you just solve and comment me your answer. If there will be any wrong, I will text you there only. Or kindly you can contact me on ubiology app. You can give your answer there in ubiology app as a chat. I will reply there, or our team member can reply you for your assistance. Now, as by solving this question, what we found, the molar mass means 1 mole molecule mass will be equal to their molecular weight. Okay, so one for any given mass of a molecule, we have to do given mass by molar mass, then you will get the mole. In coming slides, we'll see what is the molarity, molality, normality. So, if anybody is asking about this, and this is the fundamental thing by which we will get to know that if anybody asks you, okay, Ajay, you have to make you have to make 2 molar solution of 200 ml of NOH. So how you can make? So we'll see. So molarity, what does it mean? So molarity is the moles of solute dissolved in a liter of a water. Not exactly water, it can be solvent. So mostly in our labs we used water as a solvent. So mole of solute dissolve in a liter of solvent. This will be 1 liter. You can take it as a 1000 ml. So remember it, while solving the questions match the you have to match the units. So what does it mean? Molality, the same mole of solute, if you dissolve or you will mix in a kg of a solvent. So instead of taking measuring water or a solvent in liter, you have to measure in a kg, and the same mole if you are dissolving it in that kg of a solvent, that will gives you the molality. Now, normality. What does it mean? Before understanding the normality, we have to understand the equivalent number. What it is? So, equivalent number is something like suppose you have a two arrow and you have a one target. So, if you have a one target to hit, you have a two opportunities to hit a target. So, if you hit the target by one arrow, that will be one used arrow. Okay, if you didn't hit in the first arrow, so you used another arrow. So for the targeting one, for your target, you use two arrows. The same like this, you have to compare arrows as a molecules. So in this formula I can show you, there is one H2SO4, okay, and there is two molecule of NH3. So the NH3, so NH3 involved, two NH3 involved in a solution to complete this reaction. So the equivalent number of the equivalent number of this NH3 is 1, because each is involved in the two H2SO4. So H2SO4 equivalent number is what? 2. It will be equal like 1 H2SO4 is involved with the 2 NH3. Its equivalent number will be 2. Same you can see here, Ag, silver have a equivalent number of 2 and NH3 will have a equivalent number of 1. So this is all about what? Equivalent number. So that equivalent number will be given in the formula or in the bottle itself, which chemical you are going to use there. So normality will be important formula about the normality is what? The gram solute divided by the volume of solute into equivalent weight. So what does it mean, equivalent weight? Equivalent weight is the molecular mass divided by the equivalent number. Now this is a new term. What it is? Equivalent number, the same number which you seen before it is. So this is the equivalent number only, which will be which divides the molecular mass. Then it will give you the equivalent weight. Now finally you will got the gram solute by volume of solvent into equivalent weight is equal to normality. Now normality have a relation with molarity. What is that relation? So normality have a relation with the molarity. What it is? N, capital N, normality equal to equivalent number into molarity. What is it is? Molarity. Now, so there is a question which you have to solve. You just pause the button and you solve it. Now, what you have to do, you have to find the molarity. What is the formula of molarity? So, molarity is equal to the number of solute of mole. So, how many mole of solute is there? MgCl2. And water is what? So, how you can find water is a solvent or MgCl2 is a solute, or water is a solute and MgCl2 is a solvent? It is a very simple funda, and that is mostly like which is more in number, more in volume, that will be your solvent. And even though water will be always solvent, sometime for lipids or the other molecules, steroid molecules, the ethanols or ethers will be the solvent. So here MgCl2 is your solute. So mole of solute, 6 mole by 20 liter. Do we have to write in the 2000 liter, sorry, 2000 ml, sorry, 20,000 ml? We don't need, it's already given in a liter, so we will divide like this. Okay, so what will be your answer? 0.3 moles of MgCl2. So same like that, you have to give your answer in your comment box, or you can give your answer in the uBiology app as a chat. So what you have to mention, you have to mention the question as well as the answer in the chat. So this is the this is the most important thing what we have to discuss today. Before that, whatever we studied, that is the base for this all topics. So if you will see, so if you will see that what is mole, what is molarity, you seen. Now we will see, we will cover this, the calculation which is occurring in the lab. It will be like that, as I asked you, if you have to make a 2 molar solution of 100 ml of HCl, so how you can make? Or if I will ask, there is a 5 molar solution of NaCl, 5 molar solution of KOH, you have to form 5 molar solution of KOH of 50 ml. You have to make 2.5 or 2 molar solution of QH in 200 ml. So how you can make it? Okay, that you have to know. So today only we will going to read this points. So first is what? Concentration in mole per liter, we have to understand. Concentration by percentage, we have to understand. Concentration in gram per liter, it is a simple only. Gram per liter will be the concentration in per liter. And dilutions, which is very important. Sometimes we will make a stock and then we have to make a working solutions as in the SDS page and western blotting and other things. Okay, so we have to understand how to make a dilutions. Let's begin with the concentration of a mole per liter in a solution. So this formula you can derive by yourself also. I am going to show you how we can derive this formula. So you have a mole. Mole have, we seen mole have a two formula. What is it? So given mass, I am representing it as a W by molar mass. So I am representing as a how should I represent? Small m. Okay. Okay, that is a mole. So molar mass I can represent with the double m. Okay, suppose another mole formula is what? Okay, better I will write. Now, so I'll write here mole by given mass by molar mass. Molar mass. I am representing it as a small m. Now, another formula for the mole is what? Molarity by volume. Okay, so mole equal to this mole. So this complete equation 1 is equal to equation 2. If you'll rearrange this equivalent, so you have to find from a box, what? From a from a box or from a bottle or from a the crystals which you are getting, powders of a solid, powders of a chemicals which you are getting in your lab, how much you have to take from that bottles, okay, or a box. So that powder, how you can measure that is that you have to measure by given mass. Got? I hope you are getting my point. So what you have to do, you have to rearrange this equation. If you rearrange this equation, you will get this W, means how much you have to take to make a solution of 2 molar of NaOH of 200 ml or 1000 ml or 2 liters, isn't it? So you have to take, uh, you have to put W, that is unknown, we have to calculate, is equal to molarity into volume in liter into molar mass will give you the what, will give you the how much weight you have to take. That will be in the gram, not in the milligram or in the milliliter, sorry, all in the kg. So that will be in the gram. So this is all about what, the calculation. You'll see how we can calculate. So remember it, the volume will be in the thousand, it's like 1000 ml or in liter. If it is 200 ml or 500 ml, 300 ml, so you have to convert it into the liter. So if I will say there is a 300 ml, how much liter it will be, you have to just divide by 1000. So it will be 0.3 liter, it will be 0.3 liter. So hope so you understand how to make a conversion also. That is the most important thing. And if you will not understand this conversion, that will be gives you big difficulties in the problem solving. Now the second thing is what, the second thing is concentration of percentage. So concentration of a percentage, what is the use of concentration of a percentage? In your lab, some maybe your PhD guide or your maybe your PI will ask, can ask to make a 35% of a glucose or 20% of a glucose or 2.5% of glucose. What does it mean? It is a weight by volume. If I am saying weight by volume, means the weight, the chemical or the solute you have to measure in a gram. Okay, so solute you measured in a gram and divided by volume of a solution in ml into 100. That will give you the percentage volume. So you have to make it 35% of NaCl. So you can make in anything, like you can make in 100 ml also, you can make in 200 ml also. So 35% can be of 100 also, 35% can be of 200, 35% can be of 500 ml also. So it have to be just 35%. So this is easy calculation. Another is what if the chemical or the uh, chemical is not in a powder, it is in a liquid form. So what you have to do, you have to measure a volume. So solute in a ml by volume of a solution into 100, that will give you the percentage volume, percentage of volume by volume. Okay, so this both percentage weight by volume and percentage volume by volume both will be altered by the temperature. So for these things we have to do in a particular temperature in a hood, we required me. Okay, so particular temperature is required to do this thing, this type of making solution. But if you want to make a solution which is irrespective of the temperature, then you should do what, you should do for the another thing, that is a percentage weight by weight. So here, just before going to the next topic, we have to understand how to make a solution. So once you will measure how much gram of a solute you required or how much ml of a solute you required. So I am telling you for this both term, like first and second, for concentration in mole per liter or concentration in by percentage. Then if you'll measure it, then what you have to do, you have to first take a complete solute and dissolve in the half of the solvent. whatever the solvent required to dissolve the to make up of your a solution, just take half of that solution and completely dissolve that solute. Either it may be in the liquid or either it may be in the solvent. Then you make up the volume up to the limit whatever you required. Okay, this is the way. So another is what percentage weight by weight used because cannot much altered by the temperature. So percentage weight by weight will be not altered. As I can show you, as the temperature will increase in the volume by volume or weight by volume, the volume may increase. So that will alter, but weight by weight volume percentage will not alter. So this is the concentration dilution, which is very important. Okay, so concentration dilution is like if I have to make 2 molar 1 molar of 100 molar 100 ml of solution from 2 molar of 50 ml of solution. So how much ml I have to take from here, and how much water I will add? So that is your question, how much water. So this is your first tube, this is your second tube. So this is the what you have to calculate, how much concentration we have to take from the first. So so suppose this is your stock solution, this is your working solution. So from 2 molar of solution, you have to make 1 molar of solution of 100 ml. So you use this formula, C1 V1 is equal to C2 V2. C is what? Concentration. That will be in molarity, and V is what? Volume. That will be in the ml or whatever you took. If you have to make a from 1000 in liter to ml, you have to convert the volumes as per your choice. So suppose 2 molar. This is a C1 concentration, and V1 volume. I don't know how much I have to take. What? I don't know how much I have to take. So if you don't know how much you have to take, then okay, then you will write it as it is V1. So don't confuse it with the 50 ml. So this is the 50 ml of the stock solution. I don't want this complete solution. We cannot take complete solution of 50 ml. Na? We need just a some part of a solution, like some part of a solution. So what you did, how much part I need, that is given as a V1. Now how much molar I want, 1 molar, and how much ml I want to make, I want to make 500 ml. So by suppose I want to make we can take it as a uh, wait, 500 cannot be possible from 50 ml, so I am changing it little bit. So I have to make suppose 100 ml. How much I have to take? I have to make uh, 10 ml. Okay, suppose 10 ml I have to make. So what I will do, I will multiply with this. 500. Wait, something happened wrong. No. So this is the calculation. Okay, finally what you will get? You will get the 50 V1 is equal to 10 by 2, and volume you require around, okay, 5 ml you have to take. Okay, you have to take 5 ml of stock, and you raise this volume till 10 ml. You have to raise this volume till 10 ml. Then your solution will become 500. Then your solution will become 1 molar of solution. Got it? So what if I have to make 100 ml? What if I have to make 100 ml? Okay. What if I have to make 100 ml? So suppose if I want to make a solution of 100 ml of 1 molar of solution, I will multiply here as a 100, and then 100 by 2, what will you will get 50 ml. So from stock I will take complete stock because it's only 50 only. So I will take this complete stock and I will add remaining 50. So should I add directly remaining 50? No. First I will add 25 ml and I will dissolve this solution, and then I will raise till the till 100 ml. Okay. Because it is already in liquid, so there is preferred to first dissolve and then raise the volume up to your limit. So this is the way how we can make a solution in your lab. So there are some question you can try, that preparation of 500 ml of 0.5 molar HCl from 2 molar solution of HCl. So you pause this video and try to solve this question. So this is the question. So for question number 1, I am going to tell you the solution. So question number 1. Then in question you have to find which is the C1 and which is the C2, which is the volume. So remember it, mostly the higher molarity will be the C1 concentration. So this is the higher molarity, means this is your C1. Mostly higher molarity will be C1. Okay, now every time higher molarity will be the C1. Okay, and this is the, by reading this what this 500 ml of 0.5 molar means this is the volume 2. Okay, this is the C2. So here is your equation, C1 into V1 equal to C2 V2. Okay, this is your equation. Now you already have C1 2 molar into V1. How much volume I required to take from stock into 0.5 into 500. So you are smart enough to solve these questions. Okay, so this will be just 255. You need 125 ml. Okay, finally this is your answer. So V1 will be equal to this. Hope so you enjoyed this video. If you have any doubt or you want to join the live classes, you can join on U Biology, and you can subscribe our channels on the Biotech universe as well. That will be helpful for the GAT B and other examination. But for the proper preparation for NEET, JEE or CSIR, ICMR, JRF or GAT BIT JAM, for that you can join here and support us. Thanks for watching till now. Jai Hind.